Okay, good morning, everyone. My name is James Polli. I'm a faculty member at the University of Maryland at the Baltimore campus. And, okay, slides up. I'm a faculty member at the University of Maryland, and along with Anna Schwendeman, I'm a co-director of the Center for Research on Complex Generics, which is hosting this event. And welcome to Advances in Physiologic-Based Pharmacokinetic Modeling and its Regulatory Utility for Oral Drug Product Development. So, as you've seen from the schedule, for those of you who are in person here in Maryland, agendas are out front, along with a couple other flyers about the center. For those of you online, good morning or good evening or good afternoon, wherever you may be. Online at complexgenerics.org is all the materials for this meeting. I will just say that one thing that's not available yet are the slides. We make those available in about 3 weeks, since sometimes it takes us a little while to put things up. Okay, so just a little bit about the Center for Research on Complex Generics. We've been in place for a couple years. I think many of you are aware of the Office of Generic Drugs, and they have a research endeavor that's been ongoing for many years. And so they instituted this center to help them do the things that they've already been doing for quite some time. And the overall goal is to, we spend most of our time doing outreach to industry, alternatives to bioequivalent studies to speed up the development process and reduce costs. I think that's obviously a major topic during today's jam-packed meeting. Lack of case studies, even though the agency is engaged and enthusiastic and are willing to work on the issues facing industry. That's something we hear a lot about, just case studies. What are examples of best practices? And then we have some representatives from Europe here. For example, need for global harmonization and acceptance of modeling approaches for approval of generic drugs across multiple agencies. And then in terms of some selected applications, in general regarding modeling and simulation, inhalation products and solid dosage forms, obviously we're talking about oral today, population PK for assessing absorption sites, fed state gastrointestinal models with extrapolation from fasted data. Again, that's another feature of today, I think. Partial modeling for limited patient populations to round out small trials. And something we hear quite a bit about is sometimes there's guidance, but people want a little more guidance when it comes to a particular class of products. Since some of these guidance, of course, are across huge, very variant types of product types. And so some products have certain features that have unique questions to that class. Okay. Just a little plug for the next meeting in a couple months here at the University of Shady Grove. Again, it will be also be available virtually on characterization of complex excipients and formulations. And then here's a schedule of what's planned for next year, including another meeting on drug-device combination products from earlier this year, one on model master file. I think this audience would be very interested in that. Another one on immunogenicity. This is I'd say one of the most common topics that we hear about for peptide and oligo type of products. Nitrosamines, we had one last year. Again, very big topic that's maybe not unique to generics, but I'd say it's one of the generics' top issues. And then global warming potential propellants. In terms of ongoing research, so CRCG, this is probably not our major function. OGD obviously has been engaged in research for quite some time. Here's a list here. I'll just mention two, though. About 2 years ago, we did have 2 research projects on model integrated approaches to demonstrating bioequivalence for long-acting injectable products. In fact, we had an entire workshop on that. Those notes are online at complexgenerics.org. And something that we finished about a year ago, project over about a year or so, evaluation of micelle colloid diffusivity to better parameterize physiologic-based pharmacokinetic models for oral absorption. Since obviously these models that we're talking about are very dependent on their parameterization. So that went into some detail about poorly soluble drug dissolution and solubilization. Yeah, my name is James Polli. Anna Schwendeman unfortunately could not be here. Visha and Jennifer are here. And for those of you online, I think you're familiar with Dana. She is virtually here. Here's some contact information, just complexgenerics.org. Okay. And again, hopefully about 2 weeks, we will have a survey coming out on generic drug industry insights, challenges, and opportunities. And of course, we'll publicize that. Thank you for all of you that are here today. Thank you for all of you that are here online. We're really pleased to have Dr. William Jusko from Buffalo to give a little tutorial on powers and problems in physiologic-based pharmacokinetic models. But before I introduce Dr. Jusko, I just want to introduce Liang Zhao. I think all of you know Liang. He will be introducing after Jusko, he'll be introducing the framework for the entire today's jam-packed meeting. I'll just say Liang, I think you all know who he is. He's the director of the Division of Quantitative Methods and Modeling at OGD. Okay, we welcome Dr. Jusko to come up. I'm not sure what to say about Dr. Jusko. He is the SUNY Distinguished Professor at the University of Buffalo. He's been there for many years. I know many of you are students of his. He was, of course, the editor of the Journal of Pharmacokinetics and Pharmacodynamics journal for quite some time. And maybe I'll just simply add that I know he's a big Buffalo Bills fan, and I'll just mention one of his awards, the 2020 APS Distinguished Scientist of the Distinguished Scientist. So thank you, Dr. Jusko, for coming.
Change one word in the title to make it a little bit more positive. Okay, I'm going to basically speak about three issues. Point to the considerable growth of PBPK modeling, provide a few insights from our own recent research, and compliment the FDA on all of its efforts in PBPK modeling. So we all appreciate that in the area of pharmacokinetics and PBPK, there's diverse approaches that we can take. Simple non-compartmental analysis, sham, mammillary types of models are kind of anachronistic in view of the capabilities of PBPK. The ultimate, of course, is the full PBPK models, but I'm also going to mention some things about the advantages of minimal PBPK models. So these models differ from being top-down to bottom-up, different degrees of complexity, different types of utilization. The origins of PBPK can be traced back to Torsten Tyrrell, who provided this diagram with blood flow, complexity of tissue distribution. So 90 years ago, the potential was recognized, but it wasn't until Bischoff and Dietrich developed computational approaches, differential equations. They were chemical engineers, and they applied PBPK modeling to data for thiopental and methotrexate. They provided the basis for the well-stirred model, the differential equation that describes hepatic clearance. Very nice review was recently published by El-Khatib and co-workers. I'm going to show a few of their slides because it indicates the nature and growth of PBPK modeling, especially in the last 20 years. So this graph is a 20-year perspective, and the growth has been immense. Right now, there's publication of about 9,000 articles that pertain to pharmacokinetics in general, and about 120 in the recent year that consider PBPK. They notice the different areas of study. They're listed here, and probably of most relevance for this meeting today is the area of formulation, dealing with oral doses, dealing with topical doses and such. And I expect that it's going to be an area that's going to grow, largely because of the influence of all of you. The types of software have considerably helped the growth of PBPK. So PK-Sim, Simulations Plus, SimSip have been responsible for a lot of the growth. but one can see that many of us, especially in academia, write our own software to deal with PBPK. My lab uses ADAPT for the models I'm going to be showing you. This modeling trends, basically, about 50% of publications come from academia. That's very satisfying for me to see that we're able to contribute to this area. Pharmaceutical industry quite a bit, and the FDA has a modest but significant number of publications. So a few comments on our work. When COVID started, there was a lot of interest in chloroquine for treating the COVID virus. So we looked at some literature data for chloroquine in rats and noticed that there was a huge range of concentrations. Serum concentrations, plasma concentrations are the lowest on the graph. Liver concentrations are the highest. So the concentration spanned a 100-fold range. So what was responsible for that, and could we capture this kind of data? Well, dealing with tissue distribution concepts is the most challenging area of PBPK. So the kind of tissue distribution model that we enacted dealt with some of the traditional binding components of tissues, but in particular focused on lysosomal trapping because chloroquine is a lipophilic weak base. And what we found was that the major reason for the differences in tissue distribution was the fact that the lysosomal content of different tissues varies considerably, producing the diverse range. The graph shows the concentrations of chloroquine that the modeling said was associated with different components in the tissue. So lysosomal trapping is the highest. Serum concentrations are fairly low. So what we learned from all of this is that there's challenges and opportunities in dealing with complex intratissue drug concentrations and actually target interaction in those tissues. And when one looks at the in silico methods, they need some improvements, need better insights. The other area we've been dealing with recently are hepatic models. There's been some controversies in the literature. The well-stirred model is the primary one used in PBPK models, but it's possible and there's opportunities to use the others, whereas there may be some advantages to one or another in describing what is happening within the liver. We've tried to improve the utilization of series compartment models because they can be very useful for dealing with transporters and dealing with zonal metabolism in tissues. But for the purposes of this meeting, I want to point out the fact that many of you will probably be using ACAT models that are found in PBPK software. And I want to remind you that between the GI tract and the blood compartment is the liver, and the modeling needs to account for whatever happens in the liver along with the GI phenomenon that are taking place. So oral drug absorption processes are followed by passage through the liver and can be handled with a diversity of hepatic models that might be useful. The third area we got involved in with COVID was metformin pharmacokinetics. We found data from 9 species and utilized a minimal PBPK model to do a combination of a meta-analysis and allometric scaling. So the clearances of metformin follow closely with renal plasma flow, as shown in the bottom center. Tissue distribution is permeability limited, but the minimal models allow comprehensive joint analysis of all of these data and turned out to be extremely useful. So we would argue that minimal PBPK models may often suffice, especially for meta-analyses, allometric scaling, and population modeling. Last part of my presentation deals with some of the things I've admired about the FDA and what's going on in PBPK modeling. So I'm going to provide some highlights of what I see going on at the FDA in terms of PBPK. Of course, the FDA reviews submissions and there's a publication that indicates the breakdown of types of submissions with PBPK. And you'll see the big blue area are drug-drug interactions that dominate. But I want to point out the orange area that says absorption and/or food effects. So I expect with activities such as we see today that this will be a growth area for further involvement of investigators and the FDA. Mechanistic modeling has great utility in a regulatory setting. The FDA has considerable institutional knowledge and experience in having dealt with a variety of issues in PBPK. And there will be a useful resource with their insights and considerable experience in handling issues pertaining to PBPK. The FDA has various collaborations that support activities that pertain to PBPK. And some of the bullets on this slide indicate involvement with the impact of food, dealing with locally acting agents, pediatric generic products, and dealing with BCS Class 3 waivers. The guidance documents that the FDA has issued are very useful. I indicate 3 of them here, a very valuable resource for those contemplating further work in any of these areas. There's different communication pathways with the agencies, both for generic drugs and for new drugs. There's ways that one can identify from these links and such of interacting with FDA scientists as people try to advance their PBPK efforts. Global harmonization should be useful in providing a standardized approach that products throughout the world will have consistency and quality. And the FDA is involved in the M13A bioequivalence for immediate release solid osseous forms, as indicated here. Lastly, we are participating in the 3rd FDA and CRCG organized workshops. So there were 2 previous meetings the previous 2 years. The 3rd meeting is taking place here. Today's October 12th. And I'm sure we're going to see a superb program with the presentations that happen the rest of the day. Thank you for your attention.
Good morning, everyone. As Jim introduced, I'm Liang Zhao. I'm the Division Director for Quantitative Method Modeling, Office Research Standard, Office Generic Drugs, FDA. Thanks, everyone, for making to this workshop, which is going to be a key event to move the goalpost of using modeling simulation, PBPK modeling in this case, to support drug development. Just before I give an overview of the workshop, I just want to thank all the CRCG colleagues and also with Dr. Bill Jusko, who has made his way to attend this important event. As he has shown in his, one of the pie charts, currently PBPK absorption model only account for 4% of the activities. However, I think 60–80% of the medications we are taking orally. So I believe the workshop will shed, even though the activity is small, but I think this workshop will really get into the value and essence that can be delivered by PBPK modeling for oral absorption. So as you can see from the slides, there are increasing applications of PBPK absorption modeling in drug development. That's good news. And as Jim mentioned, there are also a workshop in August, which generated lots of interest as well. The PBPK models has been used to assess lots of area. I want to go through the list, but today we are going to focus on 3 areas of them. Not sure if I can control the. Another, oh, okay, I got you. All right, thank you. The purpose of a workshop is to discuss the challenges, experiences, and advances for the development of PBPK absorption modeling to support the establishment of biopredictive in vivo testing, to address risks associated with extrapolation B in various context scenarios. Even though it's mainly focused on generic, but the technique and the knowledge we gained can be extrapolated to new drugs as well, I believe. For example, from the B risk from fasting to a fast state, from subjects with normal to elevated gastric pH, for BCS biowaivers, assess B in pediatrics, how to assess the risk factors, can we extrapolate B from adult to children, to potentially support ICH effort by consolidating scientific feedbacks that we will gain today and identifying new area for harmonization. The workshop topics will, as mentioned earlier, focus in 3 areas. The session 1 will be on the advances of PBPK modeling in regulatory context to support harmonization. And session 2 will be to evaluate food effect on bioequivalence supporting ICH M13A. Session 1 also includes another session about GI locally acting product, where we haven't seen much modeling activities, but it's a very key area. We also want to gather all the expert view in this niche subject. The third area is the PBPK modeling to support bioavailability and B assessment in pediatric populations. Following that, we'll ask everyone to stay who are attending the workshop in person to collect your thought and opinion. And then we are going to expect deliberations among industry, academia, and regulatory experts on the selection of the most important topics and key issues that will influence the best practice in terms of generating value using PBPK absorption modeling. And all the breakout will be corresponding to each of the 3 workshop topics that will be covered in the morning and the early afternoon. Then the targeted outcome is that with all of your participation, and we can come out with a consolidated view on the current and future opportunities for PBPK absorption modeling, and get a mutually recognized the best practice to implement these models for regulatory use. Then we can also, will kind of channel your thought and given the feedback for ICH harmonization for their current and future harmonization. I believe the guidelines being collecting comments. I think all of the meeting discussions can feedback on how to make the improvement revisions. So before I close up, I want to also announce a very important news, good news for the modeling community, where we have launched a model integrated evidence industry meeting pilot in October 1st. The mission of the pilot is to give industry opportunity for early interaction for science-driven topics using model integrated evidence approaches for bioequivalence establishment to facilitate generic drug development and the regulatory decision-making. I believe the PBPK modeling absorption topics that we discussed today will be a key component of the meeting interactions. The vision is to have a program that allows enhanced scientific communications between the industry and FDA on using a broad range of quantitative method modeling to address generic drug development issues or questions that are either out of the scope or cannot be sufficiently addressed by the existing pre-ANDA and ANDA scientific meetings. So this is the vision of that. I hope the industry and consultant, everyone can take advantage of that. This is the FDA announcement on the website. It's very easy, just Google model integrated evidence and FDA, then you will get it. And it also have the document with the detailed operational aspect. With that, I just want to thank you, and I want to give everyone thanks for your time and participation. Looking forward for your further input. And with that, I want to also introduce the speakers of the workshop. So session 1 is on advances of PBPK modeling in regulatory context to support harmonization. So we'll have a list of 7 speakers. The first 4 speakers will focus on the ICH harmonization and advances of PBPK modeling in general, and also for oral absorption. The first talk will be given by Dr. Fang Wu from FDA, from DQMM. Then followed by another FDA colleague, Dr. Mohammed Ashraf. He is a colleague from OPQ OTR. He's a laboratory chief. Then after that, we'll welcome the third European peer, Susana Almeida. She's gonna represent European stakeholders, industry, especially industry stakeholders. And she is the clinical development and safety director, medicines for Europe. When she introduced the organization to me, it's just the equivalent of AAM version of European colleagues. Then we'll welcome the fourth presenter after that who is a seasoned speaker, Tycho Heimbach. He's the senior principal scientist from Merck, from New Drug, and I will share the thought of using PBPK modeling, I believe for the biopredictive dissolution and also the specs and the best practice. He's going to show 2 cases, I believe. If my counting is wrong, then he will make an announce when he present. So I will save for the further introductions after the coffee break for the GI locally acting product, where we have another 3 speakers for GI locally acting product using PBPK modeling. So with that, I want to welcome the first presenter for today's workshop, Dr. Fang Wu.
Good morning, everyone. And the topic of my speaking today is our advances on using PBPK modeling to support BE assessment for our products. Here is my disclaimer. This presentation reflects the views of the presenter and should not be construed to represent FDA's views or policies. First, I would like to talk about the history of relevant FDA CRCG workshops. In 2021 FDA CRCG workshop, in oral product session, we discussed about using PBPK for risk assessment and BioWeaver, and also discussed launch of the PBPK biopharmaceutical guidance and apply PBPK for BE assessment. In 2022 FDA CRCG workshop, we talked about PBPK for risk assessment, PBPK to support waiver of fat BE studies, and PBPK to support BE evaluation for pediatric products. In this workshop, we will have a whole workshop talking about oral PBPK, the advances of PBPK for oral products, harmonization and support ICH M13A guideline, and relative BA and BE assessment for pediatric products. This figure shows the general PBPK modeling procedure in ANDA submissions. The model input included in vitro data of test and reference formulations and clinical data set. Then using IV and oral data to estimate disposition and absorption parameters. After verification validation, we can conduct a sensitivity analysis to identify key factors to impact in vivo performance, and then we can use virtual BE to set clinical relevant specifications for dissolution and other critical quality attributes, and simulated BE trials between R and a T product. The regulatory questions that PBPK absorption modeling can help answer in ANDA submissions include the following areas, and they all relate to BE assessment. First, using PBPK to impact of changes of critical quality attributes and setting dissolution safe space, evaluating the risk of formulation mechanism changes, and simulate in vivo alcohol dose dumping on BE impact and justify BE study design. And the PBPK can also be used to evaluate the impact of food and gastric pH on BE, and simulate the GI local concentrations, and can support waiver of in vivo studies and support BE assessment in specific populations. Today, I'm going to talk about 3 case examples. The first case example I would like to talk about is to use PBPK modeling, evaluate impact of alcohol dose dumping on BE. Some background information. These are related to a extended release capsule, and we found that there is an increased release of 200 mg test product and alcohol dose dumping study with 20% ethanol, but not for ADD study with 5% or 40% ethanol. Here is a question: whether the increased release of test product would significantly impact the systemic exposure compared to RLD, which has slower release in the medium with 20% alcohol. For this product, the PSD recommending fasting and fat BE studies and conduct a comparative dissolution testing on 12 dosage units, each of all strengths of the test and RLD product. And also due to the concerns of dose dumping for this ER product, conducting in vitro alcohol dose dumping testing in hydrochloride medium using apparatus 2 with a different level of alcohol. To address that issue, mention the question, and we can develop the PBPK model by the reviewers and to incorporate dissolution profiles at pH 1.1 or dissolution at pH 7.5 or biphasic dissolution without ethanol into the PBPK model. And we compare the model prediction versus the observed PK data of the ROD following administration with the 200 mg drug product. Then we found that the dissolution at pH 1.1 is not biopredictive, whereas the biophysical dissolution is biopredictive to the PK profiles of ROD. Then we incorporate the biphasic dissolution with 20% ethanol into the PBPK model. And we also conducted the virtual BE simulations. This simulation, as you can see on the figure at the bottom, this predicted the R product reference and the test product are bioequivalent when incorporating the biphasic dissolution with 20% ethanol. In summary, the test and RD products are predicted to be bioequivalent using these developed biphasic dissolution profiles. This is also indicated that the increased release of the test product at pH 1.1 with 20% acetone will not result in significant differences in systemic exposure compared to the ROD product. Based on the PBPK assessment, it was concluded that there is no safety concern arising from the ADD study with higher release and deemed that test formulation is adequate in this regard. The second case example is the PBPK modeling to evaluate impact of single sex on BE assessment. Some background information. Atorvastatin is indicated in both sexes to lower the risk of heart disease and stroke. Per the ROD labeling, females have higher Cmax and a lower AUC as compared to male. The applicant conducted BE studies using male-only subjects. Here is the question: can PBPK be used to support justification that BE results from male-only subjects can be extrapolated to the entire population of both sexes? The applicant submitted atorvastatin PBPK model. This was reviewed by a reviewer, and we identified some limitations, including the applicant's PBPK model was not properly developed to capture the sex differences in drug exposure, that females have increased Cmax and decreased AUC compared to male subjects as reflected in the ROD label. And another deficiency we identified is that the applicant did not conduct VBE simulations using properly developed and sufficiently validated PBPK model to support its justification of using male population for BE assessment. Then the model was optimized by the reviewer to capture sex differences in drug exposure. VBE simulations were conducted to support the risk assessment. As you can see in the table below, and with a different percentage of female subjects in the BE simulations, we can see that the Cmax or AUC TR ratio, their 90% confidence interval are all within 80 to 125%. This indicated that the risk of having different BE results is low by using male-only subjects compared to using subjects with both sexes. In summary, for this case study, a PBPK modeling and BVE simulation demonstrate BE results from male-only subjects. Studies can be extrapolated to the entire population of both sexes. The difference in study population is unlikely to affect the BE conclusion. In addition, we also conducted research. Here, I would like to give you a case example for this research highlight. Using PBPK to evaluate the impact of excipients on BE of BCS class 3 drug products. As you may know, the nitrosamine is a commonly issue in some specific drug product. And nitrosamine may increase the risk of cancer. And nitrosamine are formed by interaction between second imine and nitrosamine ion. And one approach in controlling the levels of these impurities in the drug product is using antioxidant or using the pH modifiers in the formulations. Whether this antioxidant would impact the BA and BE of generic drug product is currently being studied. It would be helpful to use in vitro studies like permeation testing and PBPK modeling to evaluate the impact of this antioxidant on BA and BE of generic drug products. Here I will briefly give you the introduction of this research project. As a proof of concept, we have utilized an oral PBPK model of ranitidine immediate release tablet for assessing the impact of excipients on the BE of a generic ranitidine IR tablet using virtual healthy subject and VBE trials. After a validation of the PBPK model, we conduct a parameter sensitivity analysis. As shown in the figure on the right, figure A, we can see with the increase of apparent permeability, the PK parameter ratio between T and R increase. VBE simulations indicated that if with the change of apparent permeability by 10%, will show 90% of confidence interval of PK parameters, like Cmax TR ratio is outside of BE acceptance criteria. This help us to evaluate the risk of adding additional excipient like antioxidant and evaluate their impact on drug exposure and BE. In addition to these three case examples, I would like to also mention about the ICHM 13A guideline recently issued in January 2023. The PBPK modeling can also be used to support these two topics, fasting and fat study conditions and pH dependency. The design of a BE study with regard to the use of fasting and fat condition depends on the dosing instructions of the comparator product, as well as the properties of the drug substance and product formulation. The rationale can be supported by PBPK modeling. And also for the pH dependency, like we would like to evaluate whether the generic drug is still BE in subjects with elevated gastric pH. The modeling, PBPK modeling and the VBE simulations may be used to further assess the risk of bio-inequivalence under this situation. To support that ICHM 13 guideline and its future implementation, we've funded by GDUFA to support this research. One is the development and validation of best practices framework for PBPK analysis for biopharmaceutical applications in support of model-informed biowaiver of fast-state BE studies for BCS Class 2 drugs. This was awarded to the University of Florida, and another project was awarded for this project, disintegration and dissolution of solid dosage forms and influence of food-induced viscosity and kinetics for BE and suitability evaluation. This was awarded to Joanna Gutenberg University at Mainz. These two projects will all have presentations in today's workshop. And we also have similar like internal research using PBPK modeling to predict the effect of food and gastric pH on BE. Based on these research, here I listed the recent publications supported by this research. You can refer to these publications for details. Here is another list of our publications. In overall summary, PBPK modeling can be used to evaluate the impact of cold dose dumping on BE study, to evaluate the impact of single sex on BE's assessment. PBPK modeling approach may also be used together with in vitro studies like permeation testing to evaluate the impact of excipients, including antioxidant on BA and BE of new and generic products, and support formulation development to address impurity and reformulation issue. And GDUFA funded research projects and conducted to use PBPK modeling to evaluate the impact of food and gastric pH on BE and support ICHM 13A guideline. Last but not least, I would like to mention, as Dr. Liang Zhao mentioned, we just recently launched a MIE industry meeting pilot program, and this provides a venue for the generic drug applicant to discuss the modeling related questions with FDA. So yeah, you can use this pathway to communicate with the FDA. With that, I would like to acknowledge the people listed here, and also would like to acknowledge the reviewers from Office of Bioequivalence and colleagues from OPQ and OCP, and also the sponsors for submitting their data in modeling, and also appreciate the publication co-authors and research collaborators. Thank you for your attention.
Good morning. I am going to talk today on developing patient-centric quality standards, specifically with respect to dissolution acceptance criteria using PBPK modeling. My disclaimer is this presentation reflects the views of the authors and should not be construed to represent FDA's views or policies. I will ground my presentation with a pharmaceutical quality statement. Pharmaceutical quality assures the availability, safety, and efficacy of every dose. Patient-centric quality standards, specifically dissolution specifications using PBPK modeling. Patient-centric quality standard dissolution specification, these are standards which are set of tests and acceptance ranges which the product quality attribute should conform to to be safe and effective. This combines in vivo studies, which include in vivo dissolution profiles and pharmacokinetic data, PBPK modeling, and dissolution testing. When these three components combine together meticulously, they can guide us to the development of patient-centric dissolution standards. So overview of my presentation starts with PBPK modeling, and everybody's expert here in PBPK modeling, which is really foundational to define a safe dissolution space and patient-centric dissolution standards. So I will explain this approach of establishing dissolution specification using one of the FDA's case study, and at the end will reflect very briefly on the regulatory consideration and end my presentation. So PBPK modeling is PBPK model defines or describes the drug concentration time profile in the human body by taking into account the dynamic interplay of various physiological processes and their effect on the PK profiles. It's interconnected of three major components, which are drug input functions, physiological compartments, and clearance processes. So drug input functions may be intrinsic and extrinsic, and then the physiological compartments includes all the tissues and organs where the drug is distributed and affects the concentration time profile in the body. And then third column, the processes which affect the profile. All these components built, and this model then can predict, learn, and confirm, and it's a continuous process. So next is dissolution. Conventionally, dissolution testing, dissolution methods have been developed with the discriminatory ability of the method in mind to guide the development of formulation and process, and afterwards to use it as a QC tool to release batches to maintain batch-to-batch uniformity and quality. But it never took into account the in vivo dissolution environment in the body and in release drug profile. So naturally, these in general, these dissolution processes were not dissolution data were never predictive to in vivo profiles. If this PBPK modeling is used in this manner, and it can define a safe dissolution space, and it can predict actually virtual by equivalence, and can be very useful in predicting, in supporting the SUPEC kind of changes. So developing patient-centric dissolution specification, the objective of this study was to compare the biopredictability of dissolution methods to develop dissolution standards. For this purpose, we selected lamotrigine extended release tablet. Lamotrigine is a BCS2B drug. This drug, lamotrigine being basic drug, is soluble in acidic environment. It is poorly soluble. It has a high permeability. So the dissolution, the absorption is dissolution rate limited. And being basic drug in the intestinal fluid, it is expected to be of converting to non-ionic form and may precipitate in case of higher doses. So we studied this drug, the dissolution of this drug in using USP2 and USP3 apparatuses under birelevant and non-birelevant conditions. And we changed the pH to maintain the sink condition. We varied the agitation rate, etc. And we changed the gastric resident time to mimic the transition in the gastrointestinal tract. And then at the end, we use this dissolution data to relate to in vivo release profile with the help of PBPK modeling. For developing the lamotrigine PBPK model, this data was included in the database, which included in vitro drug properties. For example, solubility in water, in biorelevant media, and at different pHs, pK table, etc., fraction unbound to plasma, bile salt, solubilization rate, etc. Population estimates for age-related physiology module was updated in terms of weight, age, gender, and ethnicity as taken from the clinical data and clinical studies. First pass extraction of 1.63 for lamotrigine was used, and a two-compartment PBPK model was two-compartment PK model was used using PK plus module of Gastro Plus. And this model was then verified using an IV data set for the elimination phase of lamotrigine. For this purpose, we took IV data of a 50 milligram dose of lamotrigine administered by IV infusion to 12 patients, and assuming a two-compartment model, and then we predicted the PK profile of this dose. And if you see closely this figure, the predicted profile is very closely replicating the actually observed data. So this establishes that the PBK model developed is very accurately describing and explaining predicting the elimination phase of lamotrigine. And this elimination phase we use to develop, to predict the absorption phase of oral lamotrigine doses for 60 milligram and 120 milligram tablets. And to verify that, we compared it with the actual clinical data from two different studies for this purpose. And if you see the absorption phase of the predicted profile is again very closely simulating the actual clinical data. So this establishes that the model can predict the absorption phase very accurately. Then we use this elimination phase and absorption phase from these two studies to predict the PK profile following extended release lamotrigine tablets. For this purpose, we used 100 milligram dose, 200 milligram, and 300 milligram tablet of lamotrigine. And we again verified this predicted profile using the clinical data. And as you can see, the predicted profile very closely replicates the actually observed data. So this establishes that this model can predict accurately the absorption of extended release tablets and immediate release oral tablets, etc. Then in the next step, we had to develop a dissolution profile which very closely mimics the in vivo profile of the drug, as we understood based on the physical chemistry of the drug itself and the physiology. For this purpose, we used USP2 and USP3 operators, and we used biorelevant and non-biorelevant media. We included in that surface active agents. We varied the agitation rate, etc. And we tested the dissolution of lamotrigine tablets, 50, 100, 200, and 3 milligram tablets. So here are these two tables indicate the condition, experimental condition of the dissolution media, etc. in USP2 operators. And the right-hand profiles, dissolution profiles show the dissolution of 50, 100, 200, and 300 milligram tablets in biorelevant media. These profiles clearly show that the drug release is not complete, and this is probably due to the non-sync condition because of the limited volume of, because of the non-sync condition. Then the right-hand profile shows a dissolution in non-biorelevant media, And there's no discrimination of 50, 100, and 200 milligram dissolution profile, which indicates that the sink condition was adequately maintained, and the release is nearly zero order. Then we use this in vitro profile to correlate it with the in vivo release profile. And here is a summary of the IV-IVR fitting results using this USP2 data. For this purpose, we use mechanistic, numerical, and Lou-Riegelman deconvolution functions. And green arrows shows the acceptable correlation, and the red arrows show unacceptable, inadequate correlation. And this Lou-Riegelman function shows that the correlation adequately correlated with for all the strength under both relevant and biorelevant dissolution media. Then we conducted dissolution in USP3 apparatus, where we change the dissolution media to biorelevant and non-biorelevant conditions, as well as we used surface active agent, sodium lauryl sulfate. And in this, because of this non-biorelevant media, the analytical procedure also included solid phase extraction, which has its own implication on the whole process. So the profile on the upper left-hand side shows that the release is in the biorelevant media is affected by the surface active agent. When we are using surface active agent, which is the orange profile, the release is faster and closer to 100%, while the one with the green color, where there's almost no, there's no sodium lauryl sulfate, drug release is incomplete. And at the same time, it's not close to zero order release. But in the non-biorelevant media, we change the agitation rate from 5 to 10 to 15 dips per minute in the USP3 apparatus. And this condition was able to discriminate between this profile. It has an effect on dissolution. And we use this dissolution data to correlate it with the in vivo release profile using PBPK model. And here is a summary of the results. So in this case, you see the numerical deconvolution function was able to give acceptable result, whereas mechanistic deconvolution function did not give acceptable result. It failed for Cmax, and this is because the non-sync condition was not maintained. Then we wanted to validate this IV-IVC model. For this purpose, we needed a fast and slow release formulation of lamotrigine, which we manufactured in-house. The composition is described here, and we already have the clinical data for fast, medium, and slow profiles, which we then, so for these fast and medium and slow release lamotrigine tablet, we conducted dissolution in USP2 and USP3 apparatus as under the similar condition as described earlier, both in biorelevant and non-biorelevant condition. The profile in USP apparatus 2 under non-biorelevant conditions clearly differentiate and discriminate between slow, medium, and fast release, and the release is close to 100%, as well as it is nearly zero order. Whereas in USP3, the release is not complete in the first place, and it's not close to zero order. It's again due to non-sync conditions. Then we correlated, we validated this correlation for both internal validation and external validation, for which we use Wagner-Nelson, Lou-Riegelman numerical and mechanistic deconvolution function. For internal validation, we use slow, medium, fast formulations, and we predicted the correlation of the medium formulation. For external validation, we did IV-IVC model for fast and slow and predicted the medium formulation. So here are the results of this validation. For all validations, the Cmax and AUC, passed for USP2 dissolution data under both non-biorelevant and biorelevant medium conditions. So IVIVC passed predictability criteria for both internal and external validation. The top right-hand side profile shows the point-to-point correlation of the fraction released in vitro versus the in vivo fraction released, and the lower shows the the predictability and the predicted and the actual profiles, and the AUC and Cmax both meet the criteria. For dissolution data generated in USP3, the Cmax really failed. And this is again because of the non-sync condition of the medium. So IVIVC failed Cmax predictability for both the internal and external validation. Sync conditions in the alkaline and the colon medium is the most probable reason for it. So what we learned from this case study, dissolution characteristics were more sensitive to the formulation and dissolution parameters, and IVIVC level A was established and validated. The internal and external predictability was not improved by using biorelevant media. The variability of dissolution space was identified, and patient-centric quality standard for dissolution may then be established from this information. So my reflections on the regulatory considerations has already been mentioned a little bit. Regulatory agencies acknowledge PBPK modeling potential to enhance dissolution specifications and predict in vivo behavior. The FDA actively engages with industry stakeholders, and PBPK modeling data in regulatory submission can expedite the approval process. And in the end, PBPK may be used to identify safe dissolution space to establish patient-centric dissolution standards. Setting patient-centric dissolution standards lead to improved drug performance and therapeutic outcomes. And we observed some research gaps, which are, number one, lack of complete physiological data in model predictions, understanding the impact of physiological variability on dissolution, and refining the IVIVC model to capture complexities of the drug release and absorption accurately. Addressing these gaps will improve the reliability of dissolution predictions and pave the way for precision medicine. With this, I come to the acknowledgement. Various offices participated in this exercise, in this case study, which included OPQ, the Office of Sub-Office of Testing and Research, and Office of New Drug Product, and Office of Translation Science, Office of Clinical Pharmacology, etc. And with this, I thank you for your audience, and I'll take questions later in the panel discussion.
Hello, good morning. My name is Susana Almeida. First of all, I'd like to thank the invitation to be here. It's really my pleasure. I've heard we have a big audience also online, so to those hearing us from various parts of the world, also welcome. I really like the introduction with establishing the parallel between Medicines for Europe and AAM. In fact, Medicines for Europe is indeed the generic and value-added and biosimilars trade association from Europe. I have a pretty easy role here today because I am not the technical expert in modeling, and my role here is to bring the perspective from the generic sector from Europe. I'm also a member of the ICH M13 expert working group, but I'm wearing purely my industry hat today. So there will be later on a session dedicated to that. What I would like to say is that model-informed drug development is critical for both generic and new drug development. There are various aspects, including in the concept of model-informed drug development, like PBPK that we are discussing here today, PBBM, population pharmacokinetic modeling, and model-based bioequivalence assessment. The use of these model approaches can bring access to complex and costly drug therapies and make them affordable and accessible to broader populations of patients worldwide. To this effect, ICH is developing the M15 guideline, and this is the background for that. Besides increasing the use of MIDD analysis, there's really no common understanding on the approaches for use of these methodologies within and between the regulatory agencies and industry across the world. There's also lack of documentation standards, the concepts of model validation, assessment, even the terminology requires some harmonization. And this really leads to the over-reliance on more empirical approaches, and this results in really non-efficient drug development strategies and study designs. So the scope of ICH M15 is to provide guidance on quantitative strategies, analysis, and interpretation of results, also standardization of reporting and documentation. So this provides an overall framework for the use of these model-based approaches. It introduces the concept of risk-based assessment, which is a good basis for the use of MIDD. It provides a framework for multidisciplinary teams to strengthen the interaction and dialogue involved in drug development. And it is a high-level, really, guideline with recommendations regarding interactions between sponsor and regulator, regarding the design of these studies, the planning and submission of these studies. All of what I'm saying is part of the final concept paper of M15. And this is also important to consider. The concept of model-based approaches has been around for a long time, and the utilization and the potential growing reflect the needs of generic companies. The interactions between applicants and agency offer already some opportunity. It was very interesting to recently find out about this initiative of the FDA for discussion of these model-based approaches. And I think we need this kind of initiative because this is really a new area. And the more we discuss together, the better the common understanding of what is going to be required. This increasing MIDD importance is recognized by ICH because we have this topic M15, and M15 will provide useful guideline with these general principles to model-based approaches. And this will enhance the collaboration among the generic drug companies to support methodological advancement of these approaches, including all of the angles of model-informed drug development. But ICH is not only focusing on this in the form of M15. We've discussed here today, and we will have a session afterwards about the M13, which is applicable to immediate release products. As has been discussed, M13 mentions the possible use of PBPK in 2 cases, pH-sensitive drugs and fasted and fed for high-risk products. I'm using the terminology in the draft guideline. It is possible, as with all guidelines, once the public consultation is over, that the ideas or the terms vary slightly. So this is still going to be discussed at M13 at the next steps of the harmonization and consensus building process. So this is what I meant to say, that we only have a draft guideline for the moment, and this draft guideline is expected to reach the next level quite soon. Beyond this, we have had the adoption by the ICH General Assembly of a topic on modified release products, and this is expected to start after the M13 guideline series is more advanced, and it is an important next step for harmonization of bioequivalence standards for these more complex dosage forms. Now, in preparation for this trip here, I was looking last week at PubMed, and just last week, my literature monitor showed 4 publications in reference to modeling. And it's quite interesting because in the first presentation today, we saw these very interesting data of how this is becoming an area of intense publication, which means intense scientific activities. So I pause here to say, some of you who know me know I'm a diver. That's my picture in a cave in Mexico. One of the things about cave diving is that it's really life-threatening. So we have to prepare very well. And one of the things that we know as a principle of cave diving is that we have to work together, and we have to work as a team, and we have to learn from those who are having experience in this field. So initiatives like the one we have here today, I find extremely important because it brings not only the scientists, but the regulators and industry together discussing these new topics. And this is really what is required when you have a new and exciting area like model-based approaches. So my thoughts, and we have at Medicines for Europe a group that has been spending some time discussing these model-based approaches, is that the speed of science is, of course, faster than the speed of regulation. And early joint discussions among the scientists and the regulatory agencies will help reach a joint scientific understanding on this new and important topic. What is the importance of this? It will increase predictability. And when you talk to the industry, you will often hear this, the concept that predictability is important, because if you are planning to develop a drug, you want to know what you need to do for this development to meet the requirements of the agencies. So increased predictability, that's a very important angle of this. And it's also relevant to contribute to the development of regulatory standards and facilitate subsequent potential harmonization initiatives. Harmonization doesn't occur solely at the level of ICH. I mean, before we reach maturity of a concept to reach the ICH harmonization level, we need extensive scientific discussion. We need to have some understanding that we are all on the same page, and there's really good potential for the harmonization of the topic. So I'd really like to, again, congratulate the organizers and all of you in the room and all of you who are connected remotely. This sort of initiative is exactly what I meant when we say we have to discuss this and we have to reach a common understanding, and this is a very important and significant step towards that. So I'd like to end by thanking Dr. Pavel Farkas from Teva, who's one of the IGBA's topic leaders in the M15 expert working group, and also Medicines for Europe Bioequivalence Working Group and the Model-Based Bioequivalence Task Force. Thank you very much for your attention.
Good morning, everyone. It's been a great session already, and I'm here to talk to you about PBPM. I think you can use the next slide. You will hear from me a couple of slides on PBPM versus PBPK that Dr. Jusko had already mentioned a lot about metrics on PBPK application. And I will have 2 case studies in the 15-minute time slot. This slide I'm just showing because Dr. Jusko had mentioned about the 4% in terms of absorption modeling used as part of PBPK. I'm very hopeful that in the near future, we will have double or triple that percentage in the FDA regulations, particularly, as you call it here, PBPK for absorption modeling food effect and also for drug product quality. So if you don't remember anything, I hope you remember this circle, because the big circle is the PBPK that was discussed earlier by Dr. Jusko, with various applications from the preclinical side all the way to special populations. And The reason we came up with the terminology PBBM is because it focuses on drug product quality. So if you see dissolution data, and if you see dissolution data being modeled, then it's really a possibility is more an application of PBBM, where you're focusing on critical quality attributes such as dissolution data. I will speak to you a couple of examples as the previous speakers did on the dissolution bioequivalence safe space. And at the end of August, we had a workshop here in the same room for 3 days with almost 200 attendees and over 70 regulators discussing exactly what PBBM is and how we can enhance knowledge and understanding on the regulatory side and also on the industry side, such that the model applications become more common and there's more of a global understanding what PBBM can be used for. The slides are also available on the same website as here from the University of Maryland that you can find still there if you couldn't go. Since this is a workshop focusing on generic drugs, we just would like to highlight that PBBM, looking again at drug product quality attributes, is typically more used in the late phases of drug development in contrast to the innovator companies. But on the innovator side, PBBM is really used as early as IND stage, because we would like to know whether we need an enabled formulation or not. So a lot of the PBBM modeling actually never makes it to regulatory agencies. This is again in contrast perhaps with the generic industry. And just reminding everyone, right, when we are talking about PBBM versus PBPK, you see there in the middle different dissolution profiles that are modeled with different methods. So the most commonly used ones are the Z-factor or Takano model. There's the Weibull function models, and there's also the product particle size distribution models. The reason I show this is typically when you talk about PBPK, you're talking about people who are doing DDI studies, and here with PBBM, we're talking about the modeling of these dissolution profiles. This brings me now to the first case study that is relevant to fevipiprant and also known as QAW039. This is a drug that was very late in development, and there's a lot of clinical data was available. And because the drug was discontinued, it's now possible to discuss in detail the dissolution data put together by the team shown here on the screen. You can see that fevipiprant is a selective reversible antagonist of prostaglandin D2 receptor. It was used for the treatment of asthma, and it's a BCS4 drug. It's with iron, and you can see the structure there. You can see there is C13 and N15 tracer label on it. This was used as part of the microdosing IV study, so IV parameters were available. And you can also see the dissolution model was the Z-factor on Weibull function. Regular QC method was used to generate dissolution data. And again, it's a low molecular weight drug. The purpose was to help and support dissolution specification settings. So that goes to a significant drug product quality questions. We used the GastroPlus in this case, and the multiple PK data were available. The publications will show more than what I can show here in this short amount of time. There were 150 milligram dose data and 450 milligram dose data included in the data set. And just in terms of the model setup, walk you through. It's always nice when you have IV data, because then you can check the box and regulators are happy. So here we have the microdosing data. That means the microdose was given on top of an oral dose. And that means there's no issue with dose linearity and clearance being dose dependent. And the dissolution model here, as shown, was done with the Z-factor on Weibull function. Various parameter sensitivity analyses were conducted specifically on the vibro function and the extent of release. The manuscript is very elaborate, so anybody who has never done it, the supplemental section has 80 pages to describe exactly how to do parameter sensitivity analysis. You can see the model was applied for the 150 and 450 milligram, and then this was then used in the last step to help with the specification setting. I'm only showing limited data here from the publication, which is the 450 milligram, and there's two panels. You can see on the very left is the fast vibro. This was a faster dissolution profile. And in vivo, deconvoluted, you can see in red, you have 100% release relatively quickly. Then there was a slower dissolution profile, which has a slower in vivo release. Calculated here, you can see it's only about 90%. This difference resulted in a difference in Cmax, a reduced Cmax with the slower formulation. On the bottom panel, you can see, if you just look at the scale, for the slower release, the maximum concentrations were about 4,000 nanogram per ml versus 6,000 nanogram per ml for the fast dissolution profile. So you can imagine, like, typically when we see bioinequivalence, as we see here, this is typically on Cmax. And so the slow formulation was found to be bio non-equivalent, and that's to the liking of the FDA PBPK draft guidance on oral biopharmaceutical applications, which says ideally the sponsor, when we're using such PBPM models, should have, they call it unacceptable bioavailability data. So we have non-BE for this formulation and for this drug. And here you can see in green, this is the safe space. It's replotted from the publication. We have the 400 milligram fast release on the top in brown, and then the pink curve is the bio non-equivalent formulation, which was the slow formulation. So between those, you can call that the knowledge space. But then the question is, where are you setting your specification at? So in this case, various virtual profiles were generated, and the publication spells out how they were generated. But you can see the dashed lines, there are virtual profiles, and the specifications ended up being set at 60 minute and 80% release. And you can also see that the blue line, that was the commercial product to be marketed, was very well within the safe space. I'm just going to switch over to the second case study, which is Merck drug molnupiravir. I'm talking about a poster presented last year at AAPS, and I'm very grateful to all my colleagues listed here on the poster who contributed to this work. Molnupiravir is a prodrug, it's an ester prodrug, isopropyl ester prodrug of the nucleoside analog NHC, and NHC inhibits the viral replication of SARS-CoV-2. The drug is approved in Japan. One thing which we found out is the solubility of both the prodrug and the metabolite NHC is very high. It's between about 40 to 90 milligram per ml, and those number is consistently below 1. For the permeability, we use the rat intestinal perfusion model, and you can see at the bottom the permeability calculated for the prodrug is higher than that of high permeability marcometopolol. And this is a little bit of a busy slide, but it speaks to the richness of the data that we have used in this model. There were various these are all independent studies. There was no crossover study at the time. So what you're seeing is on the left-hand side, there is oral solution data, and then there was a separate study which was capsule data. And then there was yet another study, which was a food effect study, where we had also the control arm that we used for the model setup. We do not have intravenous data for this drug, but what we do have is we have species agreement in the preclinical side to scale the clearance. And if we look in the literature, actually, because this is a nucleoside, the data are very consistent. So we felt comfortable with the allometric scaling approach, which was confirmed by population PK and also had species agreement bottom up. We had then used additional studies that we used for verification. You can see that at the bottom, these were included hospitalized adults. Again, completely independent study. And then there was another study with the capsule reference. So these are just some of the data that we have available to us. And the question was, the purpose of this whole modeling was to demonstrate that the dissolution profiles from capsules manufactured at dissolution at different manufacturing sites are bioequivalent. So this is a little bit of a busy slide here. And I'll just tell you in 1 minute what you're looking at. The blue curves with the open squares, that's the metabolite. You can see the metabolite in the lower right corner. Again, it's the decarboxylated metabolite that is measured in the systemic circulation. And on top, the top 4 panels on the top show the solution data. And on the bottom, we have the capsule data. So you can see that the fit visually is quite good. It's only selected data that we're showing here. A predictive error did not exceed 20% typically in these independent studies, and the maximum was 25%. So here you can see some of the dissolution profiles from different manufacturing sites. They are labeled C, L, and W. And we can see that we demonstrated the capsules will be bioequivalent. And since then, we have continued with formulation development, and we have also included some slower variants, and the model holds true that we have a wide bioequivalence space significantly beyond what beyond F2 criteria. And there are additional publications that we have. I think the slides are posted online. So with this, I'm going to thank everyone for listening.